All right, hello. My name is Olivia Leland. I am a physics PhD student at Brandeis University. I'm going to be talking about my research today and make this cartoon illustration that you see in front of you make a little bit more sense. In particular, I talk about biophysics and the physics of multicellularization. But before we go into what my research is, I want to give an intro into how I got to where I am today. So I actually did a culinary arts program when I was in high school. I was in a conservatory school. So half of my day was academics and half of my day was culinary arts and hospitality. I picked some of the prettiest things that I made here. I loved pastry and I specialized in baking, but I had a really enjoyable time here, and everybody was very surprised when I went into science because I think everybody was very convinced I was going to end up in the arts to some capacity. Now, one giant takeaway that I had from my education in culinary arts and hospitality is learning about food science. So every good chef is a good scientist. When you are learning how to make certain recipes and make really good food, especially with pastry, you have to know about how the science of food works. So for example, what you have here is just a cup of cream. And when I take something like this, which is a liquid, and I whisk it, I can make whipped cream, right? We've all done it, or at least we've all seen it. Now, what you see here on the left and the right is actually two different stages of whipped cream. So on the left, you have what you call soft peaks. So you see this like dip right here, and you have stiff peaks, which you see on the right. Now, from a culinary standpoint, you're going to be whipping to stiff peaks, and we'll know that because of applications. But what physically is going on here that differentiates between a whipped cream that's a little softer versus one that is a little more stiff or more solid-like? That actually comes down to some basic physics, specifically the physics of foams. So what you have here is a gradient. Let's say on the far left, you have the cream. On the far right, you have the air. The more and more I whisk that cream, the more I'm going to make these bubbles that are going to get more and more compact. So if you have a soft peak, it's going to be somewhere here. You have liquid between it. And if you have a stiff peak, they're going to be very, very close together with virtually no liquid between the bubbles. We describe this as the excluded area between these bubbles, meaning in a softer foam, a softer whipped cream, there is going to be a higher excluded area because there's going to be more area between these bubbles, versus for a really stiff foam, you're going to have a much lower excluded area that's virtually zero when you make a really stiff whipped foam. So this was kind of my first intro into food chemistry and to food science. And that's why when I went to community college, I actually majored in chemistry. So I chose to go to community college because I couldn't afford going directly to a four-year university at the time. I wanted a little bit of time to figure out what I wanted to do. And I spent my time there taking a variety of STEM classes. Again, I got my associates in chemistry. You can see here, these are actually samples of bioengineered bacteria producing different fluorescent pigments. And this is me studying some biology with my cat. I can tell by the amount of labeling that I've done. Now, I really enjoyed my time here. I actually did a research opportunity in biopharmaceuticals, but I learned that while I was very interested in chemistry and I like chemistry a lot, I wanted to pursue a little bit more of the physics because I found that very interesting. And so then I transferred to UCLA and took on a research project that has a little more physics in it. Just as a kind of brief explanation on what I did, I synthesized what are called nanoparticles that had dyes encapsulated inside of the nanoparticles. So these nanoparticles are maybe at least half as large as a human cell. So they're much smaller than human cells, and they contained a really dense concentration of these dyes. We took these nanoparticles and then we injected them into mice so that we could image them as they circulated in the mouse's body. Now, why are we doing that? The reason why we're doing that is because when you perform imaging, you're going to get different quality of images depending on where in the light range you are imaging. Specifically, we looked at infrared radiation, so infrared deep tissue technology. As an example, this is the mouse head. We are imaging about right here. So this is 850 nanometers. This is not in the visible light spectrum anymore. It's invisible, but this is what the mouse head looks like when we're right here in the infrared spectrum. Let's take a little bit more of a shift in the infrared spectrum, and you see much higher clarity with this image. You can see all of the individual brains in the veins in the mouse head. And you're going to get much better clarity, much better resolution. So again, non-visible light, but the use of this technology improved the imaging that we had in the deep tissue spectrum. I love this project. I learned so much being at UCLA. And I found that in this project, what I found most interesting was the photophysics, was learning about how this imaging technology worked, how it could find better resolution, more than maybe the synthetic chemistry aspect. And so from all of these experiences, I pivoted towards looking at the physics and biological systems. So now I'm going to give a little bit of an intro on my project in particular. So I think many of you are familiar with the difference between bacteria and eukaryotes. Bacteria are prokaryotic organisms, which means they don't have any organelles. They have this circular loop of DNA, and the DNA is basically free floating in this organism, right? It doesn't have a nucleus, unlike the eukaryotic cells, which would be like a plant cell, an animal cell. They have organisms, mitochondria, they have linear DNA, and the DNA is bound inside of this nucleus. We've learned very extensively about bacteria and eukaryotic cells in many capacities. However, there is a different type of organism that is not as frequently discussed, and that's called Archaea. So Archaea are prokaryotic cells. So similar to bacteria, they have no organelles, they have circular DNA, it's free floating, so there's no nucleus, but genetically their proteins are way more similar to eukaryotic cells than bacteria cells. To visualize this, we know that Archaea is the third domain of life. So basically you have bacteria, Archaea, and all eukaryotic cells. And while they are prokaryotes, you can see this illustration here is showing you that they are very similar to eukaryotic cells. Now, where do you find these Archaea? The ones that we look at in particular are called haloarchaea. So these are extremophiles that are adapted to live in very, very salty concentrations of water. So this here is actually a lake in Australia. This pink area is just fully filled with different haloarchaea. Haloarchaea are pink. That's why the body of water is pink. And this is regular seawater on the other side. You can also find them in Himalayan sea salt, which I think we've all seen, those pink sea salt lamps. They tout a bunch of health benefits from the ions floating in the air, but they're pink because of haloarchaea. Now, how salty are we talking about here? Just as a gauge, your blood is 0.9% salt. Seawater is 3.5% salt. And one species of haloarchaea, Halopharax vulcanii, actually thrives at 18% salt concentration. So that's why they're thriving in these really, really high salinity bodies of water, salt lakes, or even trapped inside of those salt lamps. So what in particular is very interesting about these Archaea, besides the fact that we know very little about them and there is many opportunities to discover many different things about them. So if you've seen the way that most cells divide, you have one mother cell, they pinch off and they become two cells, mitosis. And that's what you're going to see on the left here. These are Archaea that are growing, dividing, separating from a mother cell to a daughter cell. This would be a colony. We've seen this time and time again. Now, what we can also do when we image them is we take some Archaea, that's our little orange circle here, and we put them underneath an agarose pad, which is basically a jelly pad. And this jelly pad actually applies downward force and squishes them underneath that jelly pad. And so what happens when you compress them, this is one single Archaea, one single cell, grows, divides, segments, and turns into this collective cluster. Now you can see very visibly the difference between these two division cycles, but this one on the right, this type of division is a type of division that we actually only see in eukaryotic tissues. And so this is what we call clusters. And we found that when you compress these cells, they only form clusters under certain conditions. So under really low agarose concentrations, they have a lot of separateness between them, tying back to when we were talking about foams, they have a higher exclusion area. So these are probably going to be more fluid versus these are really tight together. The exclusion area is virtually zero. So these are going to be much stiffer as a whole. Architecturally, they're both very different in terms of how they pack and combine with each other. And so what we love about Archaea is they are nature's shapeshifters, and they take up a lot of different morphologies. We have the teardrop, we have what I call the mermaid, we have our clusters here, discs, we call this the Dorito. They have a lot of different shapes and a lot of different morphologies. So I have a lot of different species that I can put under the pads, squish them, and see how they react. So far, I've worked with a bunch of different species in the Halopharax genera. So these are all different Halopharax species. And the key takeaway I want you to have from the series of pictures is like the first picture that I showed you, which is Halopharax volcanii. They are forming clusters, but this forms under 1.5% agarose, and these ones only form under 2.5% agarose. What you're seeing here is the ideal concentration of agarose that yields healthy, well-behaved clusters. So below this, they're forming colonies, they have a large exclusion area, and above this, they are getting very sick, if not dying. All of these species die when we hit a concentration of agarose that's about 3.5% because it's just too stiff. However, there are some species that do thrive in environments that are very, very stiff. So this one, Halopharax lucentense, it forms under 3.5% agarose. Of the 14 different species that I've looked at, this is one of 2 species that is able to live in these very, very stiff conditions. All of the other ones die. So this is very unique and kind of begs the question, why are some of these species able to survive in such severe environments? Why do they form under different conditions so widely? And what can we do to figure out how they work? Another thing that they do is they're very dependent upon their shape. So in those previous examples I showed you, they start as discs, so little circles, and they form circular clusters. This specific species basically looks like a noodle prior to us compressing them. And when they divide in really low agarose concentrations, this is very moist, very squishy, there's virtually no pressure on them. They kind of clump together, they make this compact noodle structure, versus on the right, they've been compressed and they start clustering, but because they initially were super duper long, you see the initial shape right here, along this line right here, they form a really long, elongated, non-uniform cluster. And we don't know why this is yet, and we don't know why they react to these forces differently than the circular cells. And so the reason why we're looking at this is because we can apply the same physical concepts that we've applied to foams and also apply them to cells. So here on the left, we have 2 different major types of cells. You have epithelial cells, which are going to be like skin cells, tissue cells. You'd find them in your gut. They're very closely compact together. They have exclusion areas virtually zero. They're very stiff. I mean, if you poke your skin, you're not going to poke right through it. Versus mesenchymal cells, in a very severe circumstance, let's say this is something like blood cells, which are very dispersed, are not going to be tightly packed together when they're healthy, and they're going to be a lot more fluid. And so these are really severe cases of, very different polar opposites, but we actually see this when organisms, higher level organisms, develop. So what you see here on the right is the development of a zebrafish embryo. All of these individual dots are individually tagged cells. So each of these dots is one cell. And if you see as it's growing and develop, it's starting to specialize. Some of the cells are starting to get more compact. Some of them are starting to spread out and get a lot more further away from each other. This specific area is going to turn into the tail, and you see how densely packed these cells are. And we know with other measurements that if we measure how soft this section is versus how soft this section is, This is going to be much stiffer collectively and apply more force in the environment than the cells in this environment. They're much less packed. This is going to be a very high exclusion area zone versus a very low, closely packed exclusion area zone. So most of the research we've had into cells behaving as foams has been in eukaryotic cells, because bacteria don't do the same effect. And so now I'm hoping to translate these maths, these simulations, to the Archaea, which is something that has never been done before. And I'd like to give an acknowledgement to everybody in my lab, my advisor, Dr. Alex Bassan, and everybody who has helped and funded me in this project.